Hello? Hello? Can you guys see me yet? No. This is the continuation of my cell biology in a nutshell, um and life science notes. So we were learning about DNA, the genetic material, and currently we're learning about how DNA is turned into proteins, which is an important process that occurs. And here's a diagram showing the steps. Not sure why it's not working. It says RNA polymerase adds complementary RNA nucleotides to a template DNA strand. The formed RNA strand is identical to the other coding DNA strand, except for uracil is substituted to thymine, I believe. Nucleotides are added only to the 3' end of the RNA molecule. Various proteins bind to a sequence near the 3' end of the pre-mRNA molecule. About 10 to 30 nucleotides downstream, the cleavage and the polyadenylation specificity factor cuts the pre-mRNA at a cleavage site. So DNA, you have a template coding strand of DNA, and then RNA. The RNA nucleotides make a strand complementary to the DNA, except that U is substituted for T in the RNA strand. So then adenine, a cap and a tail are added to the 5' and 3' ends of the new RNA molecule as protection to help it to leave the nucleus. The 5' cap consists of a single guanine nucleotide. The 3' or polyadenine tail consists of hundreds of adenine nucleotides. So RNA transcript. So polyadenylation on the 3' end says about 250 A's adenines are added to the 3'. And then a 5' cap is added as well. The 5' cap makes it so that both ends of the mRNA are protected from nucleases, which target the 5' ends. Also, there are introns and exons inside. Introns are non-coding intervening sequences which are removed from the RNA strand before the RNA leaves the nucleus. The remaining coding segments of exons or the expressing sequences are joined together. The 5' cap can be recognized by a nuclear pore complex, and the mRNA leaves the nucleus. So that's the diagram. An mRNA chain is one strand of the DNA double helix in the genome as a template. The strand is called a template strand because off this RNA strand you're going to have translation, and tRNA is going to help with the building of a protein or multi amino acid chain, aka a protein molecule. Is going to be built based on the mRNA sequence. So transcription is divided into 3 stages: initiation, elongation, termination, each regulated by a large number of proteins such as transcription factors, coactivators that ensure that the correct gene is being transcribed. Transcription occurs in the cell nucleus where DNA is held and is never able to leave. The DNA structure of the cell is made up of two helixes made up of sugar and phosphate held together by hydrogen bonds between the bases of opposite strands. The sugar and phosphate in each strand are joined by strong phosphodiester covalent bonds. DNA is unzipped. Disruption of the hydrogen bonds between different single strands occurs by the enzyme helicase, leaving the single nucleotide chain open to be copied. Then RNA polymerase reacts or reads the DNA strand from the 3' to the 5' end while it synthesizes a single strand of messenger RNA and in the 5 to 3 direction. The general RNA structure is very similar to the DNA structure, but in the RNA, the nucleoside uracil takes the place that thymine occupies in DNA. Okay, get it backwards. So thymine is in DNA, uracil is in RNA. The single strand of mRNA leaves the nucleus, then through the nuclear pores, migrating into the cytoplasm. RNA which is ribonucleic acid, is used to transmit code from the DNA via codons. Codon means a set of 3 base pairs. So codons provide instructions for creating the amino acid chain, which is the amino acid, which is a building block of protein. The sequence of codons, which consists of base pairs, determines the sequence of amino acids within the protein that is synthesized. So then, let's see here. What is this showing? Okay, so this is showing translation. So, it says, let's see. So from the nucleus, the mRNA exits through the nuclear pore. So the RNA sequence is this one, mRNA, and codons are the three by 3 bases are called codons. So then this is the small ribosomal unit. Okay. The large ribosomal subunit on top. So it binds a small subunit of the ribosome, binds to the 5' end of the mRNA strand. It goes along the mRNA at the starter codon and uses the tRNA and the large ribosomal subunit is used to join the tRNA to the mRNA. So as this occurs, the tRNA which are bound, they have the anticodons matching the mRNA codons. So the tRNA which are bound to amino acids, it says there's a, through an ester bond. Is that what it says? So the tRNAs subsequently they form through the peptide bonds. The amino acids elongate into a chain. The tRNAs drop off as they're dropping off the amino acids, breaking off. And then they pick up another amino acids and bind that. So then this is using energy from ATP.